Hi friends, my topic is fermentation process, media and systems. What is fermentation? Fermentation it is a metabolic process and also there is chemical changes in this organic substrates which is present in the fermentation and this chemical changes occurs through the action of enzymes. In biochemistry, it is defined as the extraction of energy from the carbohydrates in the absence of oxygen. In the context of food production, it is defined as any process in which the activity of microorganisms brings about a desirable change to a foodstuff or beverage. The science of fermentation is known as zymology. Fermentation, as we told earlier, it is a metabolic process. Microbes' activity creates a desirable change in the food or beverage, uh, and that is like increasing the flavor, preserving foodstuffs, or providing with health benefits or any etc. The word ferment comes from the Latin verb fervere, which means to boil. But actually, fermentation is also possible without heat. Fermentation occurs in the absence of oxygen, that is, in anaerobic conditions, and also in the presence of beneficial microbes such as yeasts, molds, and bacteria, and they obtain their energy through fermentation. If there is enough sugar in the fermentation mixture, there are some yeast cells like Saccharomyces cerevisiae which prefer fermentation to aerobic respiration even when the oxygen is abundant. During the fermentation process, these beneficial microbes break down the sugars and starches into alcohols and acids and also makes the food more nutritious and preserving it so people can store it for longer periods of time and it won't be spoiled. Fermentation products provide enzymes which is necessary for digestion. Uh, for example, in humans are born with a finite number of enzymes, and these enzymes decrease with age. Fermented foods contain the enzymes required to break these foodstuffs down. So when they are decreased with age, we have digestion problems in old age, and so that is like fermentation also aids in pre-digestion. During the fermentation process, the microbes feed on sugars and starches, breaking down the food before anyone's even consumed it. So, how does the fermentation works? Microbes survive using carbohydrates such as sugars like glucose for energy and fuel. So, organic chemicals like ATP deliver this energy to every part of a cell when it is needed. Microbes generate ATP using respiration. Aerobic respiration requires oxygen, and that is the most effective way to do it. Aerobic respiration begins with glycolysis, and where glucose is converted into pyruvic acid, as we know, and when there is enough oxygen present, aerobic respiration occurs. Fermentation is similar to anaerobic respiration, that is, which takes place when there isn't enough oxygen present. However, fermentation leads to the production of different organic molecules like lactic acid, which also leads to ATP. That is, unlike respiration, it uses pyruvic acid. Depending upon the environmental conditions, individual cells and microbes have the ability to switch between the two different modes of energy production. That is, they can select or choose which mode of energy production is needed. And the organisms commonly obtain energy anaerobically through fermentation, but some systems use sulfate as the final electron acceptor in the ETC, electron transport chain. Fermentation reacts NADH with an endogenous organic electron acceptor. This is usually pyruvate, which is formed from sugar through glycolysis. The reaction produces NAD plus and an organic product, that is typical examples like ethanol, lactic acid and hydrogen gas, and often also carbon dioxide. Lactic acid fermentation. Yeast strains and bacteria convert the starches or sugars into a lactic acid, which requires no heat for preparation. These anaerobic chemical reactions, and pyruvic acid uses nicotinamide adenine dinucleotide plus hydrogen to form lactic acid and NAD+. Lactic acid fermentation also occurs in human muscle cells. We know that, right? So during strenuous activity, muscles can expand adenosine triphosphate, that is ATP, faster than oxygen, which can be supplied to muscle cells, resulting in lactic acid buildup and sore muscles. In this scene, glycolysis, which breaks down a glucose molecule into two pyruvate molecules, doesn't use oxygen and produce ATP. Lactic acid bacteria are vital to producing and preserving inexpensive, wholesome foods, which is especially important in feeding impoverished populations. This method makes sauerkraut, pickles, kimchi, yogurt, and sourdough bread. And types of fermentation, that is like we said about lactic acid fermentation. Another is ethanol fermentation or alcohol fermentation. Yeast break pyruvate molecules, and the output is the metabolism of glucose, that is known as glycolysis. The process glycolysis, in which starches or sugars are broken down into alcohol and carbon dioxide molecules. Alcoholic fermentation produces wine and beer. Mostly it is wine and beer. Acetic acid fermentation. Starches and sugars from grains and fruit ferment into sour tasting vinegar and condiments. Examples: apple cider vinegar and wine vinegar. Media which is used in fermentation are carbon sources are present, and the carbon sources is required for all biosynthesis, leading to reproduction, product formation and cell maintenance. In most fermentations, it also serves as the energy source. It is determined from the biomass yield coefficient, which is Y, an index of the efficiency of conversion of a substrate into cellular material. The next one is molasses. We use pure glucose and sucrose are rarely used for industrial scale fermentations, due to mainly because it is expensive. So molasses, which is a byproduct of cane and beet sugar production, is cheaper and therefore is used as a source of sucrose. Molasses are concentrated syrups or mother liquids which are recovered at any one of several steps in the sugar refining process, with different names depending on the step from which it is recovered. Blackstrap molasses from sugarcane is normally the cheapest and most used sugar source for industrial fermentation. Next is malt extract. Aqueous extracts of malted barley can be concentrated to form syrups that are particularly useful for carbon sources for the cultivation of filamentous fungi, yeast, and actinomycetes. Extraction preparation is essentially same as for malt wort production in beer brewing. The composition of malt extracts varies to some extent, but they usually contain approximately 90% carbohydrate on a dry weight basis. This comprises 20% hexoses, that is glucose and small amounts of fructose, 55% disaccharides, which is mainly maltose and traces of sucrose, along with 10% maltotriose, which is a trisaccharide. In addition, these products contain a range of branched and unbranched dextrins, which is present in 15 to 20%, which may or may not be metabolized depending on the microbe used. Malt extracts can also contain some vitamins and approximately 5% nitrogenous substances, proteins, peptides, and amino acids. Next is starch and dextrins. These polysaccharides are not readily utilized as monosaccharides and disaccharides, but is directly metabolized by amylase producing microbes, particularly filamentous fungi. Their extracellular enzymes hydrolyze the substrate to a mixture of glucose, maltose, or maltotriose to produce a sugar spectrum similar to that found in many malt extracts. Sulfite waste liquor, which is derived from the paper pulping industry after wood for paper manufacture, which is digested to cellulose pulp. It is used as a dilute fermentation medium for ethanol production by S. cerevisiae and the growth of Torula utilis for feed. Waste liquors from coniferous trees contain 2 to 3% sugar, which is a mixture of hexose, that is 80%, and pentose is 20%. Alkanes and alcohols, that is N-alkanes of chain length C10 to 20, they are readily metabolized by certain microbes. Mixtures rather than a specific compound are usually most suitable. So instead of a specific single compound, we can use mixtures. So this is suitable for microbial fermentation. But their industrial use depends upon the price of the petroleum. Methane is utilized as a carbon source by a few microbes, but its conversion product methanol is mostly preferred for industrial fermentation as it presents fewer technical problems. High purity methanol is readily obtained and is completely miscible with water. Nitrogen sources. Most industrial microbes can utilize both inorganic and as well as organic nitrogen sources. In this, inorganic nitrogen can be supplied as ammonium salts, mostly ammonium sulfate and diammonium hydrogen phosphate or ammonia. Ammonia can also be used to adjust the pH of the fermentation. Organic nitrogen sources include amino acids, proteins, and urea. Corn steep liquor. It is a byproduct of starch extraction from maize, and its first use in fermentation was for penicillin production in 1940s. The extract composition of liquor varies depending on quality of the maize and the processing conditions. The extracts concentrated contain about 4% nitrogen, including a wide range of other amino acids along with vitamins and minerals. Next is yeast extracts. This can be produced from waste bakers and brewers yeast or other strains of S. cerevisiae. Alternate sources are Cluveromyces marxianus, which is formerly classified as K. fragilis, grown on whey, and Candida utilis, cultivated using ethanol or wastes from wood and paper processing. The extracts used for the formulation of fermentation media are normally salt-free concentrates of soluble components of hydrolyzed yeast cells. Peptones. They are usually too expensive for large-scale industrial fermentations. They are prepared by acid or enzyme hydrolysis of high-protein materials. Meat, casein, gelatin, keratin, peanuts, soy milk, cotton seeds, etc. The amino acid compositions may vary depending upon the original protein source. Soybean meal residues. These are remains of soybeans after it is processed to extract the bulk oil, which is composed of 50% protein, 8% non-protein nitrogenous compounds, 30% carbohydrates, and 1% oil. Next is the most important, water. All fermentation processes except SSF require vast quantities of water. Not only is water a major component of all media, but also important for ancillary services, that is like heating, cooling, cleaning, and rinsing. It is a reliable source of large quantities of clean water of consistent composition is therefore essential. Then the other important factors of water are pH, dissolved salts, and effluent contamination. Next is mineral. Microbes require certain mineral elements for growth and metabolism. Magnesium, phosphorus, potassium, sulfur, calcium, chlorine, etc. are essential components which should be added. Other are cobalt, copper, iron, manganese, molybdenum, and zinc. They should be present in sufficient quantities in water supplies and as impurities in other media ingredients. Vitamins and growth factors. Many bacteria can synthesize all necessary vitamins from basic elements. For other bacteria, filamentous fungi and yeasts, they must be added as supplements to the fermentation medium. Most natural carbon and nitrogen sources also contain at least some of the required vitamins as minor contaminants. Precursors. These are defined as substances, as we know, which is added to prior or simultaneously with the fermentation and is incorporated without which and it do not bring any major change to the molecule of the fermentation product and is generally served to increase the yield or to improve the quality of the product. They are required in certain industrial fermentations and provided through crude nutritive constituents, example corn strip liquor, or by direct addition of more pure compounds. Inducers and elicitors. As we know, if product formation is dependent upon the presence of a specific inducer or compound or a structural analog, we should provide it so that it should be incorporated into the culture medium. So majority of enzymes of industrial interest are inducible. They are often substrates such as starches or dextrins for amylase. In plant cell culture, the production of secondary metabolites such as flavonoids and terpenoids can be triggered by adding elicitors. These may be isolated from various microbes, particularly plant pathogens. Inhibitors, as we know, to redirect the metabolism towards the particular product or the product of interest and to reduce the formation of other metabolic intermediates, we add inhibitors. And it can also halt a pathway at a certain point to prevent further metabolism of the target product. Example is to employed to redirect metabolism is sodium bisulfite. Oxygen and antiforms. Depending on the amount of oxygen required by the organism, it should be supplied in the form of air containing about 21% oxygen or occasionally as pure oxygen when requirements are particularly high. These oxygen requirements may vary depending upon the organism used and also the carbon source used. For most fermentation, air or oxygen supply is filter sterilized prior to being injected into the fermenter. And also another important is antiforms, which are necessary to reduce foam formation during fermentation. Forming is largely due to media proteins, as we know, which have become attached to air broth interface where they denature to form a stable foam skin that is not easily disrupted. Next is SSF and SMF, two types of fermentations, two methods, techniques of fermentation used. That is solid state fermentation and submerged fermentation. So solid state fermentation. This process occurs in the absence or near absence of free water. It is another method used for production of enzymes which involves the cultivation of microbes on a solid substrate such as grains, rice and wheat. Natural raw materials like carbon source such as cassava, barley, wheat bran, sugarcane are used. Microorganisms are selected as one of the key factors for improved yields of the product. Bacteria, yeast, filamentous fungi can be used. filamentous fungi shows better result, and also substrate plays an important role in determining the growth of microbes, thereby increasing the product yield. Substrate is chosen such a way that it should provide physical support as well as nutrients to the growing culture. Applications: solid state fermentation is being employed in various fields ranging from pharmacology to bioremediation, covering various aspects of biodiversity conservation. Production of industrial enzymes, production of biopesticides, and in bioleaching and in bioremediation. Submerged fermentation. The next technique, it is in this the substrate is used for fermentation is always in liquid state, and it contains the nutrients which is needed for the growth. The fermenter which contains the substrate is operated continuously, and the product biomass is harvested continuously from the fermenter, and the product is then filtered, centrifuged, and then dried. It is a method of manufacturing biomolecules in which enzymes and other reactive compounds are submerged in a liquid such as alcohol, oil, or a nutrient broth. It is used for a variety of purposes in industrial manufacturing. That is, their applications is submerged fermentation or liquid fermentation utilizes free flowing liquid substrates like molasses and broths. It is suited for microbes such as bacteria which requires high moisture. And also, an additional advantage is that the purification of products is easier. So that's all about my topic. Thank you.